Welcome to the Human Architecture series. In this clinical theater, we deconstruct the advanced biocomputational grids that govern human physiology. We bypass the macroscopic structures you observe with the naked eye to expose the baseline physical chemistry operating beneath the surface. This is the microscopic reality driving acute bedside survival and defining the limits of modern diagnostic testing. Step into the cardiac intensive care unit. You are standing over an unstable patient whose cardiac monitor abruptly degrades into a twisting, polymorphic ventricular tachycardia. This is torsades de pointes. The patient's myocardial repolarization has spiraled out of control, and the rhythm is lethal. You require an immediate explanation for the electrical collapse, so you pull up the patient's routine metabolic panel to check their electrolyte status. The laboratory node displays a serum magnesium concentration of 0.75 millimoles per liter. To an uninitiated clinician, this metric sits comfortably within the low normal reference range, entirely ruling out a mineral deficit as the cause of the arrhythmia. Relying on this single normal value is a catastrophic error. It pulls the clinician directly into a phenomenon pathologists classify as the Magnesium Iceberg Illusion. Circulating blood serum, the fluid that we draw for standard laboratory testing, does not reflect the body's actual mineral inventory. The normal number on the glass pane is actively masking a profound, deep tissue depletion that has completely destabilized the myocardial conduction gates. Trusting an isolated serum metric without understanding the systemic compartmentalization of this element leaves you completely blind to severe intracellular starvation. Before you can intervene to stabilize the patient, you must master the hidden transport kinetics, the biological hoarding mechanisms, and the highly volatile pre-analytical laboratory vulnerabilities of this specific alkaline earth metal. Formally designated as a divalent alkaline earth cation, magnesium possesses an exceptionally small unhydrated ionic radius, paired with a massive charge density. This dense positive charge dictates its entire biological behavior. Look closely at this structural comparison. When magnesium enters a biological aqueous environment, its aggressive charge density rapidly pulls the polar ends of water molecules toward it. It surrounds itself with a rigid, tightly bound hydration sphere, roughly 400 times larger than its actual unhydrated core. This massive watery shield completely alters its mobility. Magnesium cannot slip through standard monovalent cellular ion channels. The hydrated volume is too bulky. To cross a cell membrane, it requires highly specialized, structurally distinct transport proteins that can physically strip the water coat away, push the naked ion through, and allow it to rehydrate on the intracellular side. Once inside the cell, magnesium functions as a Lewis acid and an obligate cofactor. Observe this ATP molecule. The chemical bonds of the polyphosphate chain are packed with negative charges, violently repelling each other. The magnesium ion docks directly with the negatively charged oxygen atoms, neutralizing the electrostatic repulsion and folding the ATP molecule into a highly precise, stable conformation. Human enzymes cannot cleave or process naked, free-floating ATP. The Mg-ATP complex you see here is the true fundamental bioenergetic substrate required to power over 300 vital enzymatic reactions, from glucose processing to cellular replication. Because magnesium powers the cellular engine, the body hoards it with extreme prejudice. A standard adult male holds a total inventory of roughly 24,000 millimoles, or 24 grams, of elemental magnesium. This chart breaks down the precise compartmental distribution of that total mass. An absolute 60. This single percent is heavily guarded, restricted to a razor-thin reference interval between 0.75 and 1.00 millimoles per liter. Modern medicine relies entirely on drawing blood to test this fragile 1%. Attempting to gauge whole-body magnesium health from a standard serum test is mathematically equivalent to measuring the volume of a vast internal ocean by looking at a single drop of water. This is the exact architectural layout that creates the iceberg illusion. The body prioritizes that tiny 1% in the blood because extracellular magnesium performs a continuous life or death balancing act. Throughout the human architecture, it functions as a natural calcium channel antagonist, dictating the threshold for electrical firing at the nerve endings. On the left of this molecular map, you see a central nervous system synapse. At resting membrane potentials, a magnesium ion acts as a voltage-dependent plug, physically wedged inside the pore of the NMDA receptor. This block prevents unregulated calcium from flooding into the neuron, protecting the brain from rapid calcium-mediated excitotoxicity. To ensure this extracellular concentration never drops, the renal system operates a relentless hoarding mechanism. Unlike sodium or potassium, magnesium completely bypasses standard reabsorption in the proximal convoluted tubule. Instead, the thick ascending limb of the loop of Henle serves as the master physiological gatekeeper, reclaiming The final 10% of the filtered load is finely tuned in the distal convoluted tubule via active transcellular transport through specialized TRPM6 gates, adjusting the exact amount returned to the blood based on real-time systemic requirements. If this single ascending renal limb fails to drag the mineral back into the bloodstream, that 1% drops. The NMDA receptor plugs fall out, and the patient's entire neuromuscular and cardiac electrical stability disintegrates into tremors, seizures, and arrhythmias. A severe deficiency does not operate in isolation. Low magnesium acts like a virus, systematically breaking the body's other critical electrolytes. Observe the right side of our previous molecular map, the renal ROM-K channel. Intracellular magnesium acts as a natural inward rectifier, sitting like a lid on the channel to keep potassium safely inside the body. When magnesium levels drop, this physical block is removed. The channel is disinhibited, resulting in unchecked, massive potassium wasting directly into the urine. This is refractory hypokalemia. Simultaneously, severe hypomagnesemia chemically paralyzes the parathyroid chief glands. It entirely blocks the release of parathyroid hormone, shutting down the endocrine signal required to mobilize calcium from the bone reservoirs. The patient now suffers from concurrent plummeting calcium levels. This triad of crashing electrolytes is frequently triggered by a common over-the-counter clinical saboteur, long-term use of proton pump inhibitors, or PPIs, taken for standard acid reflux. PPIs work by brutally suppressing stomach acid, which neutralizes the pH of the entire intestinal tract. The massive TRPM6 absorption channels in the gut require a highly specific acidic environment to fold into the correct three-dimensional shape. By neutralizing the acid, the PPI warps the protein structure, physically locking the intestinal absorption doors shut and inducing severe, hidden malabsorption. When a patient presents with crashing potassium and calcium, physicians routinely attempt to correct the deficit with intravenous electrolyte bags. These interventions will completely fail. The potassium pours straight out the unblocked renal trapdoors, and the calcium resists absorption. The physician cannot fix the secondary imbalances until they replace the foundational magnesium pin first. The inverse pathology, flooding the system with too much magnesium, is rarer. Hypermagnesemia is almost exclusively an iatrogenic complication triggered by advanced end-stage renal failure, where the kidney's excretory clearance collapses and the mineral accumulates to toxic levels. Because magnesium is a calcium antagonist, an overdose acts as a severe systemic depressant. As shown at this neuromuscular junction, excessive magnesium floods the presynaptic space, heavily crowding the voltage-gated calcium channels. It physically blocks calcium from entering the nerve, entirely halting the release of acetylcholine vesicles. The downstream muscle fiber is deprived of its signal, leading to the absolute loss of deep tendon reflexes and progressive flaccid paralysis. You do not attempt to extract the magnesium to reverse this cardiac and respiratory depression. You drown it out. The direct physiological antidote is immediate intravenous calcium gluconate. You flood the bloodstream with so much exogenous calcium that it violently outcompetes the magnesium at the receptor pores, shoving it out of the way to instantly restart electrical signaling. Returning to deficiency states, the protocol for replacing magnesium demands strict bedside discipline. For stable patients, clinicians must adhere to a rigid rate-gated ceiling, infusing no more than 2 grams of magnesium sulfate per hour. Exceeding this limit forces a sudden chemical hypermagnesemia, inducing severe vasomotor collapse and hypotension. But critical care requires overriding parameters when survival is threatened. In the presence of active torsades de pointe, the physician must bypass all infusion speed limits. The protocol demands a rapid intravenous push of 1 to 2 grams over 1 to 5 minutes, violently forcing the myocardial repolarization gates shut to abort the lethal arrhythmia. Understanding the physiology is irrelevant if the data informing the physician is false. Inside the laboratory fortress, drawing and processing blood for a magnesium test is mechanically and chemically volatile. It is an absolute minefield of pre-analytical errors. The first major vulnerability is mechanical. Routine phlebotomy variables, using a small needle gauge, pulling the plunger too aggressively, or shaking tubes too violently, exert sheer physical stress on the collected red blood cells. This mechanical stress physically shreds the delicate erythrocyte membranes. Red blood cells naturally contain 3 times more magnesium than the clear plasma fluid surrounding them. When they rupture, a process called hemolysis, their dense intracellular magnesium reserves instantly leak out, flooding the plasma phase of the tube. The second major vulnerability is chemical. Drawing the specimen into a blood tube with the wrong colored cap completely ruins the analytical chemistry. The most dangerous offender is the lavender top tube, containing the EDTA additive. EDTA is a powerful and vicious chemical chelating agent. Its entire purpose is to prevent blood clotting by hunting down and permanently trapping all divalent cations in the sample. If a magnesium test is run on an EDTA tube, the chemical instantly binds every magnesium and calcium ion, pulling them entirely out of the active analytical measurement phase. These two errors dictate opposite, lethal consequences. Mechanical hemolysis generates a massive false spike in the data, making the patient look perfectly healthy while masking a severe, potentially fatal intracellular depletion. Conversely, EDTA chemical chelation generates a false near-zero result, triggering a massive and completely unwarranted clinical panic. In the measurement of magnesium, pre-analytical perfection is a matter of life and death. To protect patients and physicians from acting on these fake lethal results, modern clinical laboratory information systems utilize complex AI-driven delta check protocols that act as automated sentinels over the data. Because physiological magnesium levels are naturally highly stable, the algorithm constantly scans for biological impossibility. If a patient's level suddenly jumps or drops by more than 0.3 millimoles per liter within a single 24-hour window, the system immediately cuts the cord and freezes the entire workflow. The doctor never sees the number. This algorithmic halt forces a human clinical pathologist to physically pull the tube from the automated rack. They hold it up to the light to inspect the plasma for the pinkish-red tint of hemolysis, and they visually verify the cap color to rule out EDTA contamination. Once the specimen is cleared of physical and chemical booby traps, the platform executes a high-throughput spectrophotometric assay. Inside the cuvette, free magnesium ions are mixed with a metallochromic indicator called xylodyl blue. Under strictly alkaline conditions, the magnesium binds to the dye to form a purple-red coordination complex. The machine reads the absorbance at 510 nanometers, calculating the total magnesium concentration based on the intensity of the color shift. But a diagnostic mismatch can still occur in patients with severe hypoalbuminemia, like those with advanced liver cirrhosis. Because 30% of magnesium travels bound to albumin proteins, a loss of protein mass drops the measured total magnesium number, creating a false impression of clinical deficiency. The resolution is an immediate diagnostic pivot. The pathologist switches to direct ion-selective electrode potentiometry, a specialized methodology that completely bypasses the protein-bound fraction to measure only the biologically active free ionized pool. It is only through the combination of advanced algorithmic halting and highly specific optical chemistry that physicians can confidently act on these volatile metrics. When true hypomagnesemia is analytically confirmed, the physician must hunt down the exact anatomical location of the leak. Pathologists deploy a highly specific mathematical formula called the fractional excretion of magnesium, or FEMG. This equation conceptually compares the magnesium concentration in the blood against the amount being dumped into a spot urine sample, factoring in creatinine clearance to normalize the data. The output of this calculation is a direct diagnostic compass. An FEMG calculation greater than 4% definitively proves that the kidney's clotting tight junction magnets are broken. The renal system is inappropriately wasting the mineral, usually pointing directly to diuretic medications or tubular damage from chemotherapy. Contrast this with an FEMG of less than 1%. This tiny fraction proves the kidneys are functioning perfectly, desperately trying to save every last drop. This points the finger straight back at gastrointestinal malabsorption, strongly suggesting poor diet or those TRPM6 doors locked shut by proton pump inhibitors. Mastering this targeted diagnostic math is a core component of clinical stewardship. Ordering redundant daily magnesium panels on stable medical wards without an active therapeutic window is clinically unjustified. It runs up unnecessary costs and drains patient blood volume without altering management. By utilizing the fractional excretion formula, consultants can instantly pinpoint the exact organ failure on day 1, bypassing the need for endless, wasteful daily phlebotomy and accelerating the path to definitive treatment. To consolidate our analysis, we isolate the 5 structural mandates of this element. Pillar 1. Magnesium serves as the obligate structural cofactor for all Mg-ATP bioenergetic reactions. Pillar 2. It is the polyanionic stabilizer of covalent nucleic acid backbones, maintaining the precise structural integrity of human DNA. Pillar 3. It acts as the natural calcium antagonist, regulating central and peripheral neuromuscular excitability to prevent excitotoxicity. Pillar 4. Its homeostasis is the primary variable of the thick ascending limb paracellular loops, bypassing the proximal tubules entirely. And Pillar 5. In the core laboratory, it is the primary metallochromic spectrophotometric target of xylodyl blue dye, requiring strict protection from mechanical hemolysis and EDTA chelation. From the deep hydroxyapatite matrix of the skeletal bone through to the laboratory ion-selective electrode, magnesium is the heavily guarded, silent engine of human bioenergetics. This concludes the magnesium installment of the Human Architecture series.